some of you have seen Planctomycetes. These organisms typically grow as rosettes. So what do I mean by that? I suppose I have a marker here. Nope. So these organisms have stalks, and okay, let me try this again. So if you have a number of cells connected to each other by stalks, and those stalks can be long or short, you have rosettes. And these organisms have some really, really interesting cellular structure. So here's a rosette. Maybe you've seen this in lab at some point. Have to do something to try it out. These things are aerobic mesophilic oligotrophs for the most part. They've been most well studied by a guy in Queensland, Australia, who's recently retired. They're pretty common, but they're also nearly impossible to grow. And not very many people have been successful cultivating a lot of these things. They lack peptidoglycan, so they're naturally resistant to beta-lactam antibiotics. But they stain gram-positive all the same. It has to do with their cell structure, which is pretty weird. So this picture of Planctomycetes buckephii is really typical. So notice this stalk. This stalk, unlike other stalks in bacteria, is not a cytoplasmic extension with cell membrane around it. This is an external stalk. This is a bunch of fimbriae all bundled together to form an external stalk. The cell is covered by essentially plates. This is not peptidoglycan. It's some kind of glycoprotein, the nature of which is a little unclear. And these plates, cell wall plates, are covered in pits. Some of these have fimbriae out of them. The function of those pits is entirely unknown. Let's see. They also have condensed nucleoids. So in most bacteria, the nucleoid is fairly dispersed in the center part of the cell. In these organisms, the nucleoid is condensed. This is also true in the diatoxide, by the way. These organisms are kind of defined by their internal cellular structure. So here's Blastoporellula marina. Nice rosettes. We see these in labs sometimes. Here's what the cell looks like. And the picture doesn't quite do it justice, but I think you can see. They have a single cytoplasmic membrane. They don't have an outer membrane. This is the cytoplasmic membrane out here. And it's the light area, and it's covered by this cell wall. There's an internal membrane right here, the internal cell membrane, that separates the cell into two internal compartments. One of those compartments contains the nucleus and the ribosomes, and the other contains who knows what. Again, the nucleoid is quite condensed. This is sometimes called the riboplasm in here, and this is called the porellula plasma. In Blastoporellula, and actually for many of these with simple cell structure like this, the membranes are close to each other around most of the cell, but at one end it's quite large. Here's Isosphera pallida. This one's unusual in that it's filamentous or chained instead of forming rosettes. All of these organisms divide by budding. They don't divide by fission. And I didn't point it out, but should have. You see this right here, right? How does a filamentous organism bud? It's easy. Like so. These organisms contain gas vacuoles, and so float up and down in the water column. Here, the cytoplasm and the periproplasm here. Excuse me, that's not right. Yeah, this is the C is, in this case, this is the periproplasm, and this is the DNA. Here's the nucleoid right here. These are ribosomes, and so the cells, the plasm is all the way on the outside in a very thin layer, and then it bloods inside the cell like so. This is important because it demonstrates that this is not just the inner and outer membrane and the space in between, right? It's not just periplasm. Because in a typical cell, the cell turgor, the membrane that separates components and maintains the pressure of the cell, the ion gradients, etc., is the cell membrane. And the periplasm is essentially outside. This demonstrates that it's the this is the cell membrane, and that this internal membrane does not keep the physical pressure and ion gradients in place. That's the way it is. One of the concerns about this earlier on was that what you were looking at in organisms like Blastoporellula was that you were mistaking this was the cell membrane and this was the outer membrane, and this could be a little bit like thermos, right? And this is periplasm, but it's not, because this is at the same pressure as this. This is the oddball, Brocadia anamoxidans. This organism is an anaerobe, and it's an autotroph. It carries out the anaerobic ammonia oxidation pathway. So think of a wastewater treatment plant. Ammonia goes in, it's digested anaerobically. And for a long time, people assumed that ammonia oxidation was a completely aerobic process all the time. But it turns out there are this group of organisms and some archaea that do anaerobic ammonia oxidation. The reason nobody believed it could happen is because the inevitable intermediate in the pathway is hydrazine. Have I talked about this before? No? Any of you ever heard of hydrazine before? What's it used for? Yeah, it's an explosive. It's rocket fuel. They use hydrazine to start rocket engines because as soon as it hits air or anything reducing, it immediately reacts with it, and it'll react with anything. We used to use it to break DNA up into tiny pieces. Just a tiny bit of hydrazine for DNA sequencing. So how on earth can an organism use hydrazine as an intermediate in a metabolic pathway? The way they do it is that they have a vacuole in the cell where they store hydrazine, and it keeps it away from everything else. And so this organism has an anamoxisome membrane, it has an internal cell membrane, it has a cell membrane. Does this sort of thing remind you of any kind of organism, cellular structure you're used to hearing about? Eukaryotes, right? It's just a coccus when you look at it, though. What's that? Yeah. You know, I don't know the answer to that, but it would certainly react. Here's the most amazing one of all, Gemmata obscura globus. This thing is a coccus. It doesn't show up very well in this picture, but you have a cell membrane, the inner cell membrane. So its periproplasm is in between. And you have these internal membranous structures. And here's the nucleoid, which is condensed. Does that look like a eukaryote? Sure does, but it's not. And in fact, this thing is clearly not homologous to the nuclear envelope. Although I'll point out that the nuclear envelope is at least generally related to the rest of the cell envelope in a similar sort of fashion, right? The nuclear envelope and the Golgi and the endoplasmic reticulum are all connected in membrane structures. But this is not a nuclear envelope for a couple of reasons. One, it doesn't have nuclear pores, it just has holes in it. So the nuclear pore complex is absolutely critical for controlling what goes in and out of the nucleus. To the even small molecules, GTP is pumped into the nuclear structure and ATP is pumped out. And the organism, eukaryotes, keep an ATP, GTP differential in the cell. These organisms obviously can't do that. This thing just has holes in it. But it is a double membrane, and it does hold the nucleoli. Also, there are ribosomes on the inside, translated. Eukaryotes don't have translation in the nucleus, do they? Actually, they do. You've never heard of it before. It's called nonsense-mediated decay. mRNAs that don't have a stop codon somehow disappear before they're pumped out of the nucleus. And it's absolutely clear that ribosomes are translating messenger RNAs in the nucleus as a proofreading mechanism to make sure that they're for quality control, right? And if that ribosome gets to the poly A tail before it gets to the stop codon, that mRNA is degraded. Probably better to think of this thing as analogous, not homologous, but analogous to endoplasmic reticulum in this organism, rather than nuclear envelope. And so here's this discussion about, what's the difference between peripheral plasm and periplasm? Is this space between the inner cell membrane and the cell membrane really just the same as a periplasm in another organism? The function of this inner cell membrane is really not clear at all. There are claims of RNA being present in it, but nobody's characterized any specific RNAs that are present in it. There are no ribosomes in it. It is clear that it's the outer membrane that is the barrier between inside and outside. It holds the trigger pressure of the cell. And the diffusion membrane is the diffusion barrier as well. So at least they're analogous, but clearly these organisms are doing something different. These aren't the only organisms that have this cell structure. I'll talk about verrucomicrobia later, and they also have this internal cell structure. And there's some other organisms that may as well. That just hasn't been looked at. Oh, what did I just do?